when identifying reservoir species in multi-host communities. So I'll just start off with a little bit of motivation. So one of the major research focuses of my lab is amphibian disease dynamics. And so we spend a lot of time thinking about a fungal pathogen known as BD. You don't really need to know the details about BD for this talk, but needless to say, over the last few years and before that as well, I've sat in on a lot of amphibian BD talks. And this trend sort of started popping up in these talks. It got me thinking about a few questions. So let me go ahead and sort of tell you this trend, and then I'll sort of motivate the questions we're going to ask in this study. So what we often do in the amphibian BD world is we're out in the field, we're collecting animals, we're swabbing them, testing them for infection. So we know sort of how infected they are and also whether they're infected or not. And we do a variety of different statistical analyses, like most of us do, to understand what's driving differences in prevalence. But ultimately, in talks, you'll often see people present data that kind of looks like this. So on the x-axis, we have 3 different species. We have green frogs, newts, and bullfrogs. Y-axis is BD prevalence. And what you see here is a sort of marked variability in prevalence among species. Now, what I noticed happening is that oftentimes this conclusion follows a plot like this. So for example, eastern newts here have relatively high prevalence, and the conclusion that follows are that newts are likely a maintenance species for BD, for this pathogen. So if you do any sort of mathematical modeling, you might be scratching your head a little bit at this conclusion. But the key thing I want to point out here is that this conclusion is coming from a single data stream, so just prevalence data, and we're making this conclusion about maintenance species. So that got me thinking kind of in general, if we think about beyond amphibian BD systems and disease ecology more generally, what data are typically used to make inference about maintenance and or reservoir species? Then if we think about this question theoretically, what data are actually needed to make claims about maintenance species? And when do these things match and when do they mismatch, right? So those are the 2 questions we're going to think a little bit about today. So let's then define conceptually what we mean by a maintenance species. So a maintenance species, very generally, is a species that drives parasite persistence in a multi-host community. A reservoir species, you also hear this term, they're not quite the same thing, and I'm going to define reservoir species as a maintenance species that's also contributing to infection in other host species in the community, right? So let's look at an example here. So say we have a host community where we have bullfrogs and leopard frogs, and I make this claim that bullfrogs are a maintenance species for BD. What does that actually mean? So what that means is that I'm invoking this implicit removal experiment that says that if I remove leopard frogs, bullfrogs on their own could maintain this pathogen in the community, all right? And so the gold standard piece of evidence for identifying a maintenance species is this type of removal experiment. But of course, in most wildlife systems, that's either logistically impossible to do or just ethically impossible to do. And so we can't really do that. So how do we go about drawing conclusions then regarding maintenance and or reservoir species? So to address this, we reviewed over 400 studies that drew conclusions about maintenance of reservoir species in the disease ecology literature. And this was led by my graduate student, Joe DeMarchi. And we really kind of looked at 4 primary categories or kind of streams of evidence that could be used to identify maintenance species. So the first one were field studies. So these are studies that are collecting prevalence data, seroprevalence data, host pathogen genetics, all observational data that is being used to draw some conclusion about maintenance or reservoir species. Also lab studies. So these are going to be challenge experiments or competency experiments on species of interest and trying to draw conclusions about maintenance or reservoir species from these types of experiments. Mathematical modeling, so R0 calculations, epidemiological simulations, and also more recently, life history extrapolations. So these are going to be machine learning methods that predict out of sample reservoir species from the life history traits of species that are known to be reservoir species. So these are the 4 broad categories that we were looking at, but I want to point out that studies were often combining kind of multiple methods or multiple streams of data to draw conclusions. So what did this review actually look like? So on the x-axis here, these are going to be the 4 categories that I talked about. Y-axis is going to be the number of studies. And just sort of orient you here. So you'll see sort of multiple colors within a given column that's indicating that studies could use multiple streams of evidence. So for example, if you were a study that fell in this sort of orange region here under field study, you have used both some sort of field observational data and a mathematical model to draw inference about maintenance species, all right? So 3 major kind of take-homes from this review. So first off, 83% of studies use observational field data to draw some conclusions about maintenance species. 53% of all studies used only observational prevalence data to draw these conclusions, and 6% of field studies integrated some sort of mathematical modeling into their conclusions. So the big take home here is this isn't an amphibian BD unique thing. Observational field studies alone are really accounting for the majority of inference that we're making on maintenance or reservoir species in disease ecology. And this is happening across taxa. So if you look at host taxa, for example, oftentimes, these are the studies we were looking at, mammal hosts are the ones where conclusions are being drawn about maintenance or reservoir potential. And similarly, a lot of these studies spanned a wide range of parasite taxa with various different transmission strategies from vector-borne, direct, indirect, frequency-dependent, density-dependent. So a wide diversity in both parasite and host taxa. So this brings us sort of the second question then. So we see that observational field data, and particularly prevalence data, are used to make these conclusions about maintenance species. What are the limitations to doing this? Can this be misleading in some way? So to sort of answer the second question, we need a little more of a quantitative definition of maintenance species. And so we're going to define a maintenance species relative to the R0 of a particular species. So if R0 is greater than 1, then a species can actually act as a maintenance species in a multi-host community. And so when we make this statement that high prevalence or that prevalence predicts maintenance species, implicitly what we're saying is that I could take my prevalence and I could derive some estimate of R0 using that prevalence data by itself that would be close to equivalent to the true R0 or the true maintenance potential. Right? So we did some mathematical modeling to ask, is this generally true? Are there some specific cases that this is true? And you might not be surprised. In general, no, this is not true. So if you just have prevalence data, you're almost always going to have some sort of bias in your estimate of maintenance potential. And that bias is going to depend on community context, which I'll return to here in just a second, but also other types of correction factors. So if you have spatial heterogeneity or temporal heterogeneity or disease-induced mortality in your system, these can all bias estimates of maintenance species that you would make from prevalence data alone. Now, one of the interesting results, though, is this community context, so I'll call it like a correction factor, was actually really general across a wide range of epidemiological models. And this community context depended on 3 major streams of data. So relative host competency, relative host density, and relative prevalence. And what this tells us is that at a minimum, if you're going to be making conclusions about maintenance species from observational data, you need something about host competency, you need something about host density, and you need something about host prevalence. And that's going to be true across a wide range of epidemiological scenarios. Good news is we can actually measure these in a lot of systems. So let me just give you a quick example of how we might do this in a system near and dear to my heart, of course, amphibian BD systems. So one of the questions we're asking in my lab is who and when are driving BD persistence in seasonal amphibian communities, right? So how can we use observational data to get at this question? So I'm going to walk you through quickly 5 steps. So the first step that you just cannot avoid is model development, right? So asking a question about maintenance species is a causal question, so you need a causal model. And importantly, once you develop this model, the model structure is dictating your observational data needs. So in the case of our model here for this amphibian BD system, it's telling us that you're going to need prevalence data on all species, we're going to need relative host densities, we're going to need relative shedding rates, we're going to need spatial overlap, and we're going to need seasonality in all of these data streams. These are things we can collect in the field. But importantly, this model is also identifying data streams that we cannot observe in the field. For example, to robustly infer maintenance species in this particular system, we need something about seasonal pathogen decay rates as well. So once we have these observational data needs, then comes the model-guided field work where we're out in the field to collect these different data streams, but also supplementing this with laboratory experiments, right, to get at some of these data streams that we just cannot observe in the field. Step 4, which I won't really go into, data and model integration. This is sort of a key piece of it. But the important thing is then once we get to step 5, we have this robustly parameterized model, and we can get away from saying high prevalence equals maintenance species. Rather, we can query our model and understand how sensitive our conclusions are to parameter uncertainty and model structure, leading to much more robust conclusions about how, in this case, amphibian species are driving BD persistence. So I just want to leave you with 2 opportunities for robustly identifying maintenance species in particular systems that you're interested in. So if you're going to be using observational data, which if you're a wildlife biologist, you're going to have to do this at some point, right? Observational data is oftentimes our bread and butter, right? Mechanistic models are essential for causal inference. Asking about reservoir maintenance species is a causal question, and you can't make causal inference without a causal model. All right. You don't have to be building models on your own. You can collaborate. And there's also plenty of off-the-shelf models that can be used to help integrate and identify data streams that are needed. The second key piece of kind of opportunities or advice is that largely independent of your model, know that you need at least 3 data streams. You need at least some dimension of competency. You need some measure of host density. You need some measure of prevalence to make conclusions about mate and species. If you're doing it without these 3 streams, just keep in mind your conclusions could potentially be quite biased. So with that, I'd love to take any questions and just acknowledge some co-authors on this study and then some funding.